So next we have Kelly DeWeese and Melisa Osborne from the University of Southern California. They are PhD candidates at the university in the second half of their degree. They're working in the same research group together, and they've come up with a really nice collaboration where they're working together, looking at using giant kelp, so the seaweed with, actually they have a really nice picture, as a new biofuel. And so Melisa focuses mostly on the microbiome, where Kelly is interested in the host, the seaweed itself and the genetics, but also the metabolism of the seaweed. And so I was really excited when I heard that we could have both of them come and talk, because you can see how the two working together gives like a really nice hologenomic project by looking at the different aspects. So take it away.
Cool, thank you so much. So before we get started, a quick thank you to the webinar organizers for putting this series together, and also to Shelley for reaching out to Kelly and I and asking us to participate. We're excited to be here and talk about some of the work we're doing. So my name is Melisa Osborne. Kelly, do you want to go ahead and introduce yourself too?
Yeah, my name is Kelly DeWeese. We are both members of the NewsGene Lab, and I'm, as Shelley mentioned, going to be talking more today about the metabolism of seaweed towards domestication. And Melisa, if you want to go ahead.
Cool. So our lab overall is working on the domestication of giant kelp. So our talk today is going to be an introduction to this domestication project and how we're applying metagenomics and metabolomics to improve seaweed as a crop. So the system that we work with is Macrocystis porifera, or giant kelp. It's a type of brown macroalgae or seaweed found along coastlines all over the world. But today we're going to be focusing on the species found along the northeast of the Pacific Ocean, mainly around Southern California. So on the right here, this green line is showing the general zone where giant kelp can be found on the West Coast. So here in California, why do we care about giant kelp? Well, it forms dense kelp forests like the ones that you saw in the first slide, which is home to many important species in our local marine ecosystems. This includes sea otters, which is probably my favorite animal, and this little orange fish, which is the California state fish known as a Garibaldi. But looking beyond nature, seaweeds have a huge impact on commercial products and many processed items that we buy. One product that you've probably come into contact with is alginate, and it's a natural carbohydrate or biopolymer that can be extracted from seaweeds. It's used as a binding or thickening agent in many things we use daily, like medications, cosmetics, foods, ice cream. But what our lab is mostly interested in is developing giant kelp as a biofuel feedstock. So seaweeds can go through different refinement processes to extract alcohols or biogases. And there's several groups that are looking at how to optimize this biofuel extraction. Our lab isn't directly involved in this work, but we focus on domesticating giant kelp for commercial interests like this. The goal is to make it a high yield crop that performs well in offshore farms so that we don't disrupt natural kelp forests. So I grabbed this figure from a review by Bologna Europa. It highlights the benefits of investing in giant kelp as a biofuel resource. So it's one of the fastest growing organisms on Earth. It grows up to 18 inches a day. It doesn't compete with agricultural crops for land or freshwater resources. Its biochemical properties are a great base for biofuel extraction. It takes very little to grow, and it's full of other nutrients and biomolecules, so it's a great sustainable crop to invest in. So thinking about domestication efforts and what our lab is working on, this paper came out a few years ago from our collaborators at the University of Wisconsin-Milwaukee and University of California, Santa Barbara. And they were looking at the genetic diversity of giant kelps around Southern California. So when we think about crop domestication, it's important to have enough genetic diversity in your seedlines so that you can produce a robust crop. And these are generally saved in what are called seed banks. So kelp reproduces by releasing spores that eventually develop into what we call gametophytes. And if you compare this to land crops, you can think of these as male and female seeds. So the team at UCSB led these gametophyte collections from Santa Barbara, the Channel Islands, Carlsbad, and Leo Carrillo, which are all geographic regions within this red box that you see around Southern California on the right. And this collection is currently maintained as a seed bank by the team at UWM and more recently at USC. So the goal of the project, old school crop domestication normally takes hundreds or thousands of years. So what our group is trying to do is use sequencing and genome association tools to make this happen in 3 to 5 years. So you start with hundreds of genetically unique individuals, crops, you track their yield, and the point is to breed high-performing crop lines. So our team created what's called a common garden experiment where one male from the Channel Islands was crossed with 500 females from the remaining populations. And we created an offshore test farm in Santa Barbara that had a total of 2,500 kelp individuals. So from the original goal, which was to figure out how the kelp genome is impacting high yield growth, knowing that several factors are involved in host phenotype is what inspired our group to look at this through a more holistic lens. So quick review of what I'm talking about here. We know from studies in agriculture that host phenotype is jointly regulated by the environment, the microbiome, host genetics, and gene regulation. So this was a great figure from a recent review by Wani et al. Each color here is representing a hypothesis on how these pieces are co-regulated. So the main point of this slide is we're really not working in a vacuum, and it can be very challenging to isolate these factors. So what's exciting about this project is that we have this test farm with a ton of genetic and phenotypic diversity grown in the same location. So we're effectively minimizing this environmental variance. Our lab is taking a closer look at each of these remaining factors, and we can work together and use this approach to develop a holo-omics breeding program for giant kelp. Kelly put together this great figure as an overview of all the aspects that our team is working on. So Kelly is going to talk a little bit more about the metabolomics and RNA-seq portion of this, and I will be right now focusing more on this metagenomics piece. So when I say metagenomics, I'm referring to what's called the collective genome of the entire microbial community that's found on giant kelp. So the main questions we're interested in are who is there? So what type of bacteria is present? And what are they doing? So what kind of function are they serving? And this term holobiont is describing the host and its associated microbiome as really a unified system with an emphasis on the microbes that are truly essential to the host. Seaweeds are great models for studying associations between hosts and their microbiomes because these associations are required for normal growth and the metabolic functions of both host and the microbiome are tightly intertwined. So this is a figure from a seaweed review a few years ago, and it's a great depiction that shows how diverse these communities are and the exchange of nutrients and metabolites between seaweed and bacteria, which Kelly will talk more about. So the type of work that we're doing hasn't been applied to farm seaweeds, but we can take several lessons from land agriculture. So I think this figure is helpful to think about. The key lesson here is that there's natural variation of host phenotype and microbial communities. So if you take a closer look at the microbiome of these high-performing crops, you can identify bacteria that are differentially abundant. And then you can basically develop ways to increase the abundance of what you assume to be beneficial microbes in your seedlines. This can look something like creating an inoculant, almost like a probiotic, or even selectively breeding for kelps that naturally recruit these beneficial bacteria. So when we harvested the first round of kelps from our farm in 2019, we also collected over 5,000 microbiome samples from the adult sporophytes. We also sequenced our gametophyte seed bank to get kelp DNA so that our team could build a kelp genome, and we discovered a large amount of bacterial DNA. So what we've been working on recently is classifying these microbiome samples from these two stages, and right now we've been mainly focused on the gametophyte stage. So how we are applying this metagenomic work. This graph on the top is showing a range of biomass yields from the farm, and you can see what a wide range it is, which makes this a really fun data set to work with. We're looking for associations between individual microbes and high biomass. And since we're focused on the gametophyte stage right now, it's really seeing if you can predict output based on the microbiome from your seed stage, which is a really exciting topic to be working on. On the bottom here, this is an example of a network analysis. And the point of this is to look at how the community, the microbial community, is interacting and what microbes tend to co-occur together. So this will be helpful when thinking about how the community may respond if we introduced a novel inoculant. So as we have been searching for associations between microbes and high biomass, we have discovered that there are a few species of bacteria that are differentially abundant. We're looking into what functional niche these bacteria fill, which will hopefully inform the mechanism of how they may be contributing to increased biomass of kelps. We're hoping to start some inoculation studies soon so that we can move from predicted impact on growth to actual impact on growth. But there are a lot of directions to go with this data set. I'm happy to discuss some of the preliminary findings in more detail. And we're very open to collaboration. So if anyone is interested, please feel free to reach out. But at this point, I will go ahead and pass it on to Kelly.
Awesome. Thanks so much, Melissa. So today I'm going to be discussing the applications of metabolomic analyses in seaweed aquaculture and domestication. So kind of that other half of taking the metagenome and the host genome. So for any who are unfamiliar, metabolomics is the study of the suite of small molecules or metabolites present in an organism. And metabolites are often involved in biological reactions that are catalyzed by an organism's proteins. And so they kind of fill in the biological pathways that are occurring in every cell of your body. And studying metabolites can reveal unknown biological processes with, perhaps new metabolite discovery, or quantify metabolites of interest. And it can also designate certain biomarkers as markers for phenotype. So the thing that I want to introduce first is how metabolomics can improve agriculture before we go into aquaculture, because there's a lot more precedent in agriculture, although metabolomics applications are still fairly new. So basically, concerning this project, we're interested in one specific example that I found from a really great study published this year. I hope to do justice to it as I speedrun through these figures. But basically, it's a maize salt tolerance study, and they used untargeted metabolome profiling. So basically just seeing what's there without sort of trying to quantify any certain metabolite. And so these untargeted studies are really interesting to me because they can reveal a lot in non-model organisms. And they do this for a few hundred inbred maize lines grown in and out of salt stress conditions. And they're basically attempting to find metabolite biomarkers for tolerance to salt-induced osmotic stress. And so they use, which you can see in this bottom left figure, a random forest classification model. So they basically train it with metabolomic data for a variety of tolerance to salt stress. And they find 37 metabolites that are strong biomarkers for tolerance. And they went on to use these 37 markers in a metabolite GWAS study, so the genome-wide association study, where they're basically seeing how the metabolites are corresponding with certain SNPs, and they find basically the genetic basis of these biomarkers and investigated that further to find salt tolerance maize variants. So that's a really great example of using basically this untargeted metabolomics approach to sort of learn a lot about sort of what's going on in an organism, and also detect biomarkers of interest for specific phenotypes. So in agriculture, these traits like salt tolerance, drought tolerance are desired for like a lot of crop cultivars, but in seaweed, we're going for basically high yield and high accumulation of biofuel-relevant metabolites. So looking at this sort of a lot figure here, it's immediately apparent that metabolomics is essential for biofuel targeted domestication of giant kelp. So on the left, we have basically every process possible to create biofuel from seaweed. There's a wide variety of methods, and they rely on high composition of certain macromolecules, as well as, in some cases, extremely high biomass with or without wet weight. And so this, you can go to the next slide, Melissa. Looking at the suite of genetic, gene expression, and metabolic variation data from the farm that Melissa introduced earlier, the test farm of 500 giant kelp test crosses, we can perform basically a similar metabolomic study to what Liang et al. Did in maize, in which we would comb the metabolic data for biomarkers associated with high performing kelps. So high performance here would be, high accumulation of biomass or sugar content or fatty acid content. And very briefly, the random forest model I mentioned earlier is definitely one way of modeling metabolomic data, but something that is near and dear to my heart is this flux balance analysis sort of modeling, where you can use actual concentration of metabolites as a constraint to model biomass growth. And even in diatoms, which is a sort of a related lineage to kelps, in diatoms, we see that you can actually model the partitioning of biomass growth into lipids and sugars and the like with this type of analysis, which is one of the coolest things about this sort of research to me. And so next slide is just kind of wrapping together this whole talk and revisiting the teaser that Melissa showed earlier, which is that metabolomic analysis can be extremely interesting when it's paired with metagenomic studies. And viewing seaweed as a holobiont organism made up of a concert of host microbiome interactions begs the question of basically what exchanges are actually taking place to necessitate such a relationship. And most of that answer is in metabolite exchange. So on the top left here, just a quick look at the 16S ribosomal RNA sequencing of the associated surface microbiome to seaweed blades shows that the composition of the microbiome is specific to species and season. And basically this trend can best be explained by the existence of these surface metabolites that the algae is producing, in addition to other factors like the content of the surface cells, etc., that attract and recruit certain types of bacteria in a fairly robust manner. And you can see it's sort of a rough pattern. This is a n=2 for the seasons, but you can see that the as seasonally on the same species. So we have 2 for the same species on the left and 2 for the same species on the right. we get sort of a similar recruitment of microbes. So conversely, there is now decades of research that supports the necessity of associated microbial communities to seaweed development, especially in microscopic stages of seaweed development. So for example, without a bacterial-derived metabolite, thalicin, I don't think I've ever pronounced that, species of the green algae ulva don't develop thallus structure correctly as they grow, and you can see that pretty well in E. So those little nodules should be expanding into kind of a leaf shape. But when they're grown exenically with no bacteria or without treatment with thalicin, they're unable to form these structures. And so it's clear that the associated microbiome of seaweed is not only important to its performance, but it's also essential to seaweed survival. Okay. And then I just kind of want to wrap up with this, sort of leaving you with how we can use metabolomics along with metagenomics to sort of pick apart what pathways are working in concert in the seaweed holobiome. And understanding these relationships will enable, at least in our case, efficient domestication, we hope, of giant kelp and superior selection for phenotypes of interest, because we're understanding sort of these other effects that are going on due to the associated microbiome, but also understanding concentrations of metabolites of interest and things like that. So we have so far assembled a genome called genetic variants from our farm crosses, performed GWAS. This isn't just us, this is our whole team. And Melisa has performed metagenomic classification and association analysis with some of the farm samples. And so some of our future work is going to focus on annotating the kelp genome, which is going to be really important for these types of analyses, and as well as analyze gene expression and sequence more microbial and metabolomic samples. Yeah. Cool. All right, well, thank you all for listening. Do we have any time for questions?
Hi, yeah, thank you so much for that, Kelly and Melisa. We do have time for a couple quick questions now, but thank you so much. It's so interesting to see like the variety of topics that this can be applied to. The center is so geared towards vertebrates, so it's just really nice to see it applied in plants and also insects as well. We do have a couple quick questions, which I can just start reading out now. One is, how much of an influence would aquatic microorganisms or the water microbiome of the sea have on the success of kelp domestication?
It's a really great question. So this is something I'm currently working on. I'm still trying to figure out best ways to transform and normalize the data and how to test associations with biomass. What another member of our group has done is genome-wide association studies between kelp genome and biomass, and she's done some predictions on to what extent biomass would be increased. So this is something I'm hoping to explore. I haven't had a chance to do so yet, but it's a great question.
Okay, maybe following on from that actually is a good one. Do you think that maybe host genome factors are conditioning the microbiome?
Yeah, absolutely. So two ways that we're going to be looking at this. So one, there's a concept in ecology known as zeta diversity, and it is looking at community assembly and how that changes over physical distance between sample sites. What I'm hoping to do as sort of a justification or first step before doing genome-wide association studies is looking at genetic distance between kelps instead. So since they were all grown on the same farm, so then we can calibrate how the community is assembling over genetic distance. And what we've seen is that it actually depends if you're looking at the more rare members of your microbial community or the more common members. So we see that at larger genetic distances, the more rare members of your community are what is impacted, versus there seems to be some core microbiome among these farm domesticated seaweeds that is not impacted by genetic distance. So the next step after this is doing genome-wide association studies to see if we can find causal SNPs associated either with increased beta diversity of your microbial communities or even abundance of single taxa.
Okay, thank you very much. I think we have time for one more quick question, which is, how do you know if the metabolites are strictly produced by the organisms and not by other organisms that are in the ecosystem?
Yeah, I can take this one. So part of the gene annotation, not only in the host genome, but also hopefully of some of the metabolomic or metagenomic data, is that you could sort of pick apart what the genomes actually contain, like if they have the genes to produce the metabolite that you're investigating. And the other thing to sort of validate that would be transcriptomic analyses of most easily in the host, but basically just to see which genes are actually being turned on and being expressed. And so with that sort of analysis, we would be able to hopefully narrow down, you know, who's contributing the metabolites. And there's also some really cool stuff that you can do sort of in situ with tracking carbons and things like that that are a little more high level, but would be really interesting. Also, I wanted to answer one question. We have done whole genome resequencing of all of our farm samples. That's where the analysis is coming from.